Hello, and welcome to the ISMB 2022 Regulatory and Systems Genomics poster session. My name is Pyaree Mohan Dash, and I'm here to introduce you to MPRAsnakeflow, a Snakemake-based framework for MPRA data analysis. Now, before I tell you what MPRAsnakeflow is and how it works, I want to start by answering the question why we made MPRAsnakeflow. Every gene in living organisms is regulated by its proximal promoter elements and also by its long-range regulatory elements residing in the non-coding part of the genome. Now let's consider this gene as our gene of interest. In that scenario, nucleotide variations in these long-range regulatory regions are often found to be deleterious to the gene of interest function. It's been a decade now that we know from genome-wide association studies that more than 90% of disease-associated single nucleotide polymorphisms, or SNPs, are located outside of coding region. Now, with that information, there has been a rise in high-throughput experiments with functional readouts, such as massively parallel reporter assays, or MPRAs, that take in candidate regulatory elements, or CREs, from these untranscribed regions and introduce variants into them via mutagenesis and tag each unique elements with barcodes and create a library out of it, and further test these libraries by expressing them in multiple cell types and analyzing the regulatory activity by comparing the reference and alternate sequence of its genomic contents. Now, in this entire pipeline, there are two key steps that require high precision. They are CRE and barcode association check and the CRE activity check, both of which involving sequencing, and this is where MPRAsnakeflow comes into play. Now let's take a look at what MPRAsnakeflow is. MPRAsnakeflow is an MPRA sequencing data processing pipeline that can create count tables of RNA and DNA for each CRE involved. It follows a step-by-step process and performs count-specific statistics at each step and provides all analysis in the end as a report that is easy to export and share. When experiment replicates data are provided, MPRAsnakeflow can compute correlation among these replicates with visualizations, thereby addressing the reproducibility of these MPRA experiments. We have made MPRAsnakeflow fast and adaptable, which means it's compatible with multiple type of MPRA experimental setups, such as episomal or lenti-MPRAs. And finally, it is open source, as you might have already noticed. The GitHub link and documentation link are shown below. Now that we know what MPRAsnakeflow is, let's have a closer look at how it creates count tables by showing how it does this with the count workflow. Now, once the sequencing step is done, MPRAsnakeflow requires certain files. For example, the RNA, DNA sequence data, and experimental summary data. Let's say there are 3 replicates are done. MPRAsnakeflow needs to know that, which files belong to which experiment. A design file that has the sequences of the CREs, an optional label file if positive negative controls are provided, and an assignment file that has the mapping of the barcodes and the CREs, and the most important config file, which has all the user inputs, such as the barcode length or the unique molecular identifier length, and other configs like file locations and experimental setups. Now, once MPRAsnakeflow is run, it is able to create multiple plots, such as the distribution plot of the observed barcode coverage for each CRE in every replicate, and also the correlation of the normalized log2 ratio between the RNA and DNA also among the replicates, and a normalized CRE activity for every CRE involved based on the log2 fold change. Now, these analyses not only promise what they has to provide, but they also help in understanding the MPRA experiment better. How, you ask? Because of the nature of how MPRAsnakeflow performs its statistics, which is the step-by-step process, it enables the program to identify possible bottlenecks. For example, if we look at the end correlations, which is usually in Pearson correlations, and we see that there is a low DNA correlation at 0.14, at this point, we can say that there is a DNA bottleneck, but that's not good enough for the experimentalist. which is why we can go a step back and look at the barcode correlation, which can go either ways. And let's say the barcode correlation is actually good at 0.88, which finally means that there's an issue in the DNA library preparation. Now that's helpful to an experimentalist, and this is how MPRAsnakeflow assists in helping understanding the MPRA experiments better. Now, with our experience with handling MPRA data, we have been adding multiple features to our existing program, such as the association workflow, which handles the assignment of barcodes and CRAs, and a sampling workflow that helps us generate synthetic data to simulate a real-life experiment for statistical analysis purposes. In future, we intend to make MPRAsnakeflow a more sustainable and adaptable bridge between MPRA experiments and comparative analysis tools like MPRA Analyze. That brings us to the end of this session. This work wouldn't have been possible without the help of the members from the Computational Genome Biology Lab at Berlin Institute of Health at Charité. My hearty thanks to our group leader, Professor Martin Kischer. A special thanks to my postdoc colleague, Max Schubach. They were the minds behind this MPRAsnakeflow program. To find more about what we do and who we are, please check our website at kischerlab.github.io. The MPRA links are mentioned here again. And for any queries regarding MPRAsnakeflow, please scan the QR code for a direct email option. Thank you very much for your time, and I wish you a pleasant day.